Hello viewers, welcome to my channel Biology Notes. Today I am going to discuss about induced mutation by different chemicals. Induced mutations occur when an organism is exposed either deliberately or accidentally to a physical or chemical agent known as a mutagen. A mutagen is a physical or chemical agent that permanently changes genetic material, usually DNA, in an organism and thus increases the frequency of mutations. Let's see the main mutagens. Basically, important mutagens fall under any one of the following category: chemicals, radiation, environmental mutagens, and transposable genetic elements. In this video, I will discuss about different types of chemical mutagens. Here, I have shown main chemical mutagens. They are classified mainly into three categories: base modifying agents, base analogues, and intercalating agents. Base modifier may be deaminating agent, for example, nitrous acid, hydroxylating agent, for example, hydroxylamine, alkylating agent, for example, mustard gas, methyl methane sulfonate or MMS, ethyl methane sulfonate or EMS, etc. Base analogues mainly 5-bromouracil, 2-aminopurine, etc. Intercalating agents like ethidium bromide, proflavin, acridine orange, etc. Now I am going to discuss one by one how these chemicals act as potential mutagens. In this diagram, I have shown the role of nitrous acid as a mutagen. It is a potent mutagen that acts on either replicating or non-replicating DNA. Nitrous acid causes oxidative deamination of the amino groups in adenine, cytosine, and guanine. This reaction converts the amino groups to keto groups and changes the hydrogen bonding potential of the modified bases. Adenine is deaminated to hypoxanthine, which base pairs with cytosine rather than thymine. Cytosine is converted to uracil, which base pairs with adenine instead of guanine. From these examples, you see the deamination of adenine results in AT to GC transitions, and the deamination of cytosine produces GC to AT transitions. Thus, nitrous acid induces transitions in both directions. As a result, nitrous acid induced mutations also are induced to mutate back to wild type by nitrous acid. Deamination of guanine produces xanthine, but xanthine, just like guanine, base pairs with cytosine. Thus, the deamination of guanine is not mutagenic. Hydroxylating agent. Hydroxylamine or NH2OH is a hydroxylating mutagen that reacts specifically with cytosine. It adds a hydroxyl group so that cytosine pairs with adenine instead of guanine. Mutations induced by hydroxylamine can only be CG to TA transitions. So, Hydroxylamine induced mutations cannot be reverted by a second treatment with this chemical. However, they can be reverted by treatment with other mutagens such as 5-bromouracil and nitrous acid that cause Ta to Cg transition mutations. Alkylating agents. Alkylating agents are chemicals that donate alkyl groups to other molecules. They include mustard gas, ethyl methane sulfonate or EMS, methyl methane sulfonate or MMS, etc. Alkylating agents induce all types of mutations, including transitions, transversions, frame shifts, and even chromosome aberrations. Alkylating agents transfer methyl or ethyl groups to the bases of DNA, resulting in altered base pairing potentials. Here I have shown how MMS or methyl methane sulfonate causes mutation. MMS adds an alkyl group CH3 to the oxygen that is on 6th position of guanine. As a result, O6 methyl guanine is produced. Now, this methylated guanine pairs with thymine rather than cytosine. So, GC to AT transition occurs. Now, I will discuss about how base analogues do mutations. Base analogues are bases that are similar to those normally found in DNA. Like normal bases, base analogues exist in normal and rare tautomeric states. In each of the two states, the base analog pairs with a different normal base in DNA. Because base analogues are so similar to the normal nitrogen bases, they may be incorporated into DNA in place of the normal bases. Two best known base analogues are 5-bromouracil and 2-aminopurine. The 5-bromouracil is a thymine analog. The bromine at the 5th position is similar in several respects to the methyl group at the 5th position in thymine. In its more stable keto form, 5-bromouracil pairs with adenine. After a tautomeric shift, it transforms into rare enol form, which behaves like cytosine. So, it pairs with guanine. Now, see these two diagrams. If 5-bromouracil is incorporated in its normal state, it pairs with adenine. If it then changes into its rare state during replication, it pairs with guanine. In the next round of replication, the 5-bromouracil guanine base pair is resolved into a cytosine guanine base pair instead of thymine adenine base pair. By this process, a thymine adenine to cytosine guanine transition mutation is produced. Now, see this diagram. 5-bromouracil can also induce a cytosine guanine to thymine adenine transition mutation if it is first incorporated into DNA in its rare state and then switches to the normal state during replication. Thus, 5-bromouracil induces transitions in both directions. 2-aminopurine acts in a similar manner but is incorporated in place of adenine or guanine. Here I have shown how intercalating agents act as mutagens. Various intercalating agents like ethidium bromide, proflavin, acridine orange, etc. are potent mutagens that induce frame shift mutations. They insert themselves between adjacent bases in one or both strands of the DNA double helix. By doing this, they increase the rigidity and alter the conformation of the double helix, causing slight bends or nicks in the molecule. If the intercalating agent inserts itself between adjacent base pairs of the DNA strand that is the template for new DNA synthesis, then an extra base, for example G, is inserted into the new DNA strand opposite the intercalating agent. After one more round of replication, during which the intercalating agent is lost, the overall result is a base pair addition mutation. Here CG is added. If the intercalating agent inserts itself into the new DNA strand in place of a base, then when that DNA double helix replicates after the intercalating agent is lost, the result is a base pair deletion mutation. Here TA is lost. If a base pair addition or base pair deletion point mutation occurs in a protein coding gene, the result is a frame shift mutation. Since intercalating agents can cause either additions or deletions, frame shift mutations induced by intercalating agents can be reverted by a second treatment with those same agents. So this is all about different types of chemical mutagens and their role in mutation. If you have any query, then please comment. Share the video if you like. Subscribe the channel for more new videos. Stay with me. Keep watching. Thank you.